FUS is a nuclear RNA binding protein involving transcription regulation, RNA splicing, and transport. FUS is transported into the nucleus by carrier beta 2 or Cap-beta 2, which binds to the FUS C-terminal nuclear localization signal named P1LS. The P525L mutation and the R495X mutation in FUS P1LS cause a severe type of amyotrophic lateral sclerosis or ALS. Residue 525 is mutated from proline to leucine in the P525L mutation. A stop codon replaces the R495 codon in the R495X mutation, which deletes the entire P1LS. Both mutations mislocalize mutant FUS into the cytoplasm, but some FUS is still transported into the nucleus. We sought to understand how the FUS P525L and FUS R495X mutants interact with Cap-beta 2 and other importins for transporting to the nucleus. A crystal structure showed Cap-beta 2 binding with wild-type P1LS similar to FUS P525L, except at the mutation site where the main chain at the mutated leucine 525 shifts away from Cap-beta 2, resulting in fewer contacts and a tenfold loss of affinity. FUS R495X has no P1LS, but it still binds the P1LS binding site of Cap-beta 2. FUS R495X also binds importin beta at its importin alpha binding site. Consistently, Cap-beta 2 and importin beta can prevent phase separation of FUS. We found that the C-terminal RGG2 zinc finger RGG3 portion of FUS R495X binds Cap-beta 2 as tightly as full-length FUS R495X. This part of FUS R495X also binds several other importins. Studies of FUS R495X in cells show Cap-beta 2 as its main importin, binding to FUS RGG regions in a way that is regulated by arginine methylation. In summary, without its P1LS, FUS R495X uses its C-terminal RGG regions to bind Cap-beta 2 for entry into the nucleus, possibly allowing for partial function of FUS before disease onset.